Toxicology, vamos a hablar. Toxicology is the field of science that allows us to study the harmful effects of chemical substances on living organisms and the environment. Everybody okay with that definition? And if you have questions, please interrupt. So as I mentioned before, I arrived into the US at the peak or at the beginning of the AIDS epidemic, when the first drug to interact with the virus was developed, and it was AZT, acetate. This is a very, very simplified version of the HIV cycle, just to show you that the virus fuses their membranes with the cell membrane and injects the content. A reverse transcriptase enzyme is the one that transforms RNA into DNA, and then the DNA gets integrated in the viral DNA. The viral DNA gets integrated in the DNA of the cell, which in this case, and mostly, is a T lymphocyte. Just not any T lymphocyte, the one that has the receptors that are able to fuse the membranes of the virus. But that's not important. So the drug ACT is able to inhibit reverse transcriptase, because the reverse transcriptase is like a right hand, and the RNA comes here and the DNA meets, right? And it has a thumb domain and finger domain. And here it has the side where the ACT binds and inhibits the binding of other nucleosides. So inhibits the process. And that's very important, because that's what ACT does, to inhibit the process of the reverse transcriptase. So because when I arrived, ACT was given not only to adults, but also to pregnant women, not just to adults that were not pregnant, male and female, just to pregnant women, because HIV was transferred from mother to fetus. So to prevent that transmission, ACT was administered to the mom. So here we were in the presence of a drug that we know will get into the DNA, was given to the mom, stay in the uterus, and in a steady concentration was incorporated into the DNA of the fetuses. And what is a fetus? Besides that it's going to be a baby, it's just a group of cells that divide, and each division is an opportunity for ACT to enter. It was then that we thought we need to intervene, we need to study this. This is not okay. But what was okay, though, was that if you don't have intervention from 100 women taking this drug, 25% of them will pass the virus, but 75 will not. So it's good. It's a very good intervention. Right? But I mean, that's typical without intervention. That's what you have. HIV will go to 25%. If you give the mother ACT at any time during the pregnancy, then the number 25 goes to 4%. So that was good. That was the good aspect of ACT, and that's why doctors and the FDA were so supportive of it, because it was a good intervention. And if you add C-section, cesarean, then there is no blood connection or contamination from fetus to mother to fetus, so the reduction is to 2%. So basically, in the US, or in rural Africa, but in the US, there were no more babies born HIV positive, even from mothers that were HIV positive, because of this intervention of ACT. However, that's when the toxicologists start thinking, what is going on in the vial that is blue color, in that 96% or in this 98%? How are these children? How? Because they were born healthy, so no one will monitor them. They were healthy, they were fine, but the genetic damage, the DNA damage, you don't see it until later, mainly when it's minor, right? But it could have terrible consequences as cancer. So that's what we wanted to investigate. What is going on in those individuals that were exposed to ACT for long periods of time? And at that time, everything has a little bit of politics, but AIDS have a lot of politics. At that time, no one wanted to think about that. Because it's like now, if we say the drug that you are taking for COVID, it causes cancer. No one wants to say that, right? Well, in that moment, it was an epidemic and people were taking this and getting good results. And here we were, yeah, but cancer, they didn't want to hear that. So they told me, don't worry about that. It's a reverse transcriptase inhibitor. It's not going to get into the DNA. This is specific for the virus. Okay, well, let's look at it. And in a normal DNA replication, you have a polymerase, you have the two strands, you have the polymerase that is adding new nucleosides, right? And this is what happens, the chain grows. But when you have ACT, the Z there is the chain terminator. That means when it gets incorporated, there is no possibility of new nucleosides getting incorporated. That's why it's a chain terminator. There is a gap there. So the mix in the DNA will generate fragments of DNA of different size. And to study that, to show this effect, we did in a collaboration what is called a COMET assay. COMET assay is very nice, visually very attractive assay. To do that, what you need to do is to lysate cells, particularly the nucleus, and then you put the cells in an electrophoretic chamber in a slice. And when you apply current, the fragments will migrate to the one of the ends. And the more fragments you have, the more tail you have in the COMET. Yeah. Right? The top one is an intact nucleus. Doesn't migrate much, right? Or if it does, it's a whole thing. But in the bottom, when you have all these fragments of different sizes, you see them migrating. So this was the first evidence, and clear and no discussion about it. ACT is a chain terminator, it produces mix, and you will see comments. And then what we did is we correlated this. Because people say, how do you know this is ACT? Well, let's do a radio immunoassay and analyze incorporation of ACT into the DNA. And we did that. I developed that assay in my lab. And we correlate the tail with incorporation of ACT into the DNA. No question that this was the cause. So when you have that problem, and there are things, chances are you also need the telomeres. Telomeres are the ends of the chromosomes, and they are like caps. They are not caps, but they are protective ends. So one chromosome ends here, and the other one ends here, and they do not cross here because they have this cap, which is just a sequence and a bunch of proteins. But if you make that, then this is a sticky now, and it could exchange material with others or other end. And then when you have a mitosis, this is like a trend that is associated. It's not a normal chromosome. And this is what we see. The end-to-end fusions that are identified as chromatin bridges. And I don't have a pointer and I cannot touch, but this is one that is broken, and this is one. This is the telophase, so the nuclei are the part. This is nuclei, I'm sorry, nuclei. You don't see cytoplasm. You didn't stain for that. And then you see the bridge there, the DNA bridge. When this continues, what happens? You have pieces of DNA, the cytoplasm. And those pieces of DNA in the cytoplasm are called micronuclei. And they are direct evidence of DNA damage. And at the point that in industry, they use it as one of the factors to study toxicity of drugs that are going to be sent to market. micronuclei test is one of the ones that people do. And as you see in the first one, there is nucleus, nucleus, and then micronucleus is very clear. In that one, you have one, in the two, one over there. In that field, you have 4 already. And here you have one, which is a little bit larger, and there you have one still attached by a little strand of DNA to the main nucleus. So when you look at this, you can formulate this question. You have different sizes. That means you have different fragments. So one of the questions is, maybe the large ones have tiny chromosomes. Maybe a whole chromosome was lost. Can we investigate that? And we could, because there is an assay for that. So in order to do that, what we need to state are the kinetochores in red there. Here is a chromosome. Here is another one. This is the mitotic spindle. And the mitotic spindle for mitosis attaches, the fibers attach to the kinetochores and they pull the chromatids into the daughters. So, because we could see with antibodies, we could see the presence of kinetochores in the nucleus. We could tell if the micronucleus was negative or positive for a kinetochore. And you see on the left, a crest negative is a nucleus with different spots. Each spot represents a chromosome because it's a kinetochore. And a micronucleus on the left is empty, doesn't have a whole chromosome. And on the right, see the size? That one has 2 chromosomes. So, and in that one, you also have 2 crest negative micronuclei here and here. And this is a positive one. So again, question, like your students said today, well, no, you have more questions. Yes, you do, and that's fun. So you look at this and you said, okay, to have this problem, what do you have? DNA damage, mix, the bridges, right? And that's the what happened before. For me to see this is this mechanism. But to see that, to see chromosomes that are completely lost, that's not just DNA damage. That's something wrong in the whole mechanic of the cell division. And we need to think about what could be wrong. And in order to understand what could be wrong, we just start looking at the different structure, and these are the centrosomes. These 2 structures here that are at the end of each cell, daughter cell, and they have the command of the migration of the chromosomes, because if you look, they are attached to all of the fibers of the mitotic spindle, and they take care of that. So we thought maybe the centrosomes are known, but let's look at it. And you can also have antibodies to look at the centrosomes and to see if indeed you have 2 or multiple or 0 or what is wrong with the cell. This is my student that took care of this project. She's now a doctor for many years. So we did a confocal microscopy. Now, imaging is incredible. You guys can see physiology with imaging. This is so rudimentary. But with those figures, we got a lot of information. What we did here is we stained the cytoplasm in red with tubulin, the nuclei in blue, like you know already, and the centrosomes in green, top figure, with pericentin, which is one of the proteins that lives inside the centrosome. I'm going to show you the structure of the centrosome, but first, look at this abnormal cell. A cell should have 1 when it's in interphase, or 2 when it's in anaphase, telophase, centrosomes. No more. If you look here, these are like 9 or 10. Remember, the centrosomes live in the cytoplasm, not in the nucleus. Can you look at it, and it seems that it's in the nucleus. This is confocal microscopy. So you take a photo another one, layers, like in an onion, layers, and then you integrate, and you have a whole top to bottom. So those could be on the top or could be on the bottom, but they are cytoplasmic, okay? Because they so the cell was incredibly abnormal. And the problem with this is that it's not impaired to divide, because we did flow cytometry and we observed these cells divide like it's okay. It's not. All right. So this is the centrosome. Centrosomes are very, very interesting features. So they are orthogonal structures. They are tubulins associated structures, very similar to cilia. And they are triads of them. One is the mature, the one with appendages. They are connected at the bottom by these fibers. One is the mother, the other one is the daughter, it's the pair. And in that soup that they reside are all these kinases and proteins that determine the fate of the centrosome. And when the centrosome duplicates, because each cell at the end of the cell division has to end up with 2. So from 2 to 4, so 2 here and 2 here. And the interesting thing is that the centrosomes divide, but they don't have DNA. They are proteins, but they divide, they start growing one centrosome, and then they divide and they start a new cell. So we did study the mitotic spindle, the centrosomes, and the kinetochores, and here you can tell who is who, right? The mitotic spindle, all of these are the kinetochores. So the chromosomes in blue are migrating to the poles, and here you have our starting centrosome. This is normal, right? This is what you expect in the mitosis, the migration. This is the beta phase when all the chromosomes are in the equatorial plane. But this is like with ACT. Yeah, that's what we said too. And a control cell, of course, you have, now the colors are different, but you have, look at those centrosomes, like we say, one in each pole, and these are the chromosomes that are migrating. But each time you treat cells or with either ACT or other analogs, you have problems. You have some normal cells, but you also have very abnormal cells. But now you know who's who. The blue are the chromosomes, and the mitosis. This is mitosis, so the cell is dividing. And as you imagine, chromosomes get pulled in multiple directions, and chromosomes are lost, and a lot of DNA damage takes place. So enough information, preclinical cell biology, ACT is doing this. All the nucleoside analogs are damaging the cells in the same way, inducing DNA damage and also chromosomal, centrosomal aberrations, abnormal numbers of centrosomes. And then we said, well, okay, let's move to the mouse. We need to move with something. And mainly addressing the mother take the drug, baby gets the damage, right? So what we did is we engaged in a mouse model in which we gave the mice, pregnant mice. Do you like my interpretation of pregnancy?
Yeah.
So we gave pregnant mice ACT during the last week of gestation. Because in the humans, the last week of the last trimester of gestation is when the organs fail, and they don't need to develop anything neurologically or so. So it's less risk of teratogenesis, right, or malformations. And then we just gave it in the last week of gestation to the mom per mouse, and then we allowed the little pups to be born, and 1 year later, we studied the incidence of tumors in both arms, the treated arm and the control arm. And what we found was tumorigenesis, a lot of tumorigenesis in every organ that we look, in every tissue that we look, except hematopoietic tissues. there is a whole story about that, very interesting one, hematopoietic went down. So, in the reproductive organs of the female, in the lungs of male and females, and in the liver, you have a clear dose response in which the control, which is the clear yellow, had a level, but then you have an increase, up to 57% increase in the liver of the male. And that was not good news. And no one liked it. And the newspaper cited us before we could publish. CNN cited us before we could publish. And people went down. But anyway, this was what happens, right? And so what we did is what we wanted to do is, can we look at the blood of these individuals, the mom and the fetus, and know how much incorporation of DNA, of ACT into the DNA took place before they develop the tumors, right? They might develop the tumors 1 year at 1 year of age, which will be equivalent to a 20-year-old person, more or less. So if we know that in the blood of this individual, because of pharmacology, because of genetic makeup, incorporates more ACT into the DNA than others, we can measure it. I mean, we can do things. We can do some prevention, right? So we developed this test. We gave ACT, the same regime, but now we sacrifice the animals at birth, and we pull all the livers of 1 liter, right? And we extract the DNA, and we study incorporation of ACT into the DNA of these newborns. And we studied incorporation, mitochondrial DNA and nuclear DNA. And we find very interesting things. First of all, the mitochondrial and nuclear are very different compartments with different enzymes, different incorporation, lack of repair for mitochondrial DNA in some cases, so there's sometimes more incorporation. And that different organs have different levels of incorporation. And also look at skin. 100, the top there, 100 molecules of ACT per million of nucleotide. That was the highest value, right? In animals that develop cancer 1 year later. So naturally we went to humans. I'm putting the 20 years here for slides, right? But we went to humans. So what happens in humans? And in humans, we're not going to take a liver, right? So you are going to take. And what we did is we took blood from mother and baby at birth, from umbilical cord and from the mother, from the peripheral blood. And what did we do? We analyzed ACT incorporated into the DNA. Do humans incorporate? They do. Do they incorporate is there a relationship between mother and baby? No, look at the second and third column. The babies that were exposed for only less than a month, if they were 3/4 of a month, they had very high incorporation, much more than the mother. There are some, like 4 months, in which probably the mother never took the drug, or she couldn't metabolize because no baby or mother never had anything. And also there is no relationship between time of exposure to the drug and incorporation. It's worse at the beginning because you don't have time to develop resistance. It's a completely different topic. But look at the amount, it's 400. 400, so 4 times more than the skin of the mice that had the highest incorporation and they had cancer. So just saying, this is what is happening. So of course, when this information was there, what we want was that now in the label is clearly stated that this drug is a carcinogen for mice, right? And that the mother is facing the terrible decision of what do I want for my kid? I want an HIV positive baby, or I want a kid that is exposed to a drug that later on in life will put him at the risk of cancer. But at least is evident for the medical system, and it could be monitored. Okay, so that's my research. And I was very pleased with it because at some point I was doing research just because I wanted to know. And one day, when you wanted to know, made an impact in society, such as, wow, the drug that they are using for its therapy induces cancer. So it's like, wow, I can live now. I know I made an impact in my research that will help people. And that was very nice. And that's what you probably think is going to happen at some point with whatever research you are doing. Okay, so that's what I did with genetic toxicology. What other people do, when most toxicologists work to assess and understand how chemicals affect living systems, and they can develop mechanistic understanding of effects, right? So why I see this drug produces a headache? Because the cytokines, or because there are no endorphins, or the receptors. And so they put a name to a reason of why there is a side effect. And they ensure safer chemical products, a little bit of what I was doing, right? Develop safer drugs and medicine, actually, determine risks. So you can take 3 weeks of this drug, no more. Develop treatments for chemical exposures. They know what organ is affected. They know how to counter that action. And ensure safe food and water supply. And also, everybody's always like with forensics, because now the technology is unbelievable. Okay, and as I mentioned today, you have different targets. It could be acute, it could be reproductive toxicity tests, right? Developmental toxicity tests, ocular, and on the skin, a lot of cosmetic products use animals to test the toxicity on the skin. Phototoxicity test, that's unbelievable. I went to NCTR in Arkansas to see the photo lab they have, in which they have a room like this or larger, all with light that mimics the solar light. And they place drugs and they put them under the sun, and they see the differential effect that the sun reactivation of the drug has in different tissues or systems compared to a drug without no sun. It's very important. Then you have toxicokinetic studies prediction of what is this drug gonna do in these individuals, and behavioral tests, cognitive tests, or whatever. Oh, yeah. Sorry.
Cancel.
Okay, don't say.
Get that back on.
Wait.
Thanks.
Okay, you can go home.
No.
Just kidding. Salami.
First time I see that there could be such an abbreviation with telomeres. I didn't know that there's more than 4 on a cell.
Yeah, a lot more,
I
yeah.
have a question.
Yeah.
The chromosomes are still inside the nucleus of the
Yeah.
cell?
Yeah, but remember in metaphase, you don't have a nuclear membrane, right?
Yeah.
They are in the equatorial plane.
So that's the place where they can be used.
Yeah,
Oh, right.
there is no.
But they can still make their be useful if they're inside
Yes.
the. Okay.
They
So
are
they can be
separate.
outside the main group, but still be functioning.
They are functioning, they are probably not synthesizing at that time until they recover integrity as a nucleus in the daughter cell.
Okay.
And the whole mechanism of synthesis starts. Thank you. Okay.
She has a question.
You were talking about the molecular, with the chromosomes inside of it, which is going to activate, are they still functional? Like the agricultural.
The micronuclear?
Yeah.
Well, it's controversial because when I was studying in Argentina or so, micronuclear were just a thing there. And they were used to quantify DNA damage. That's it. But now, little by little, later, people are claiming that those pieces of DNA are active. And that, but it's not controversial, but it's just incipient research. That's a good point, because they are there, why not? And the enzymes are there, around, so, right. And also, they will get lost in the next cell division.
Oh,
So the test, micronucleus test is very interesting. You do it in the human, exposed, you take blood and you analyze it in red blood cells. Why? Because they don't have a nucleus. So you see the red blood cell, which is just like a disc with a micronucleus. It's very easy to quantify. Yeah. There is more to it. It's a very nice answer. Okay, so how does the toxicology science relate with other sciences? Well, cell biology, you really clearly I show that. When I say biology, molecular biology, biochemistry, of course, with all these kinetics and studies, chemistry, epidemiology, because like now with COVID and all these drugs and ACD, when it was given in different areas of the world, right? And genetic toxicology. And something I want to show you to finish is, this is a page I took from, information I took from a page from the Society of Toxicology, in where the Society of Toxicology is incredibly so large and it has something for everybody. You will be able to find your niche or what you would like to learn more about it. And it has, because it's so large and not all the audience is at the same place, on the same field, they have what they call specialty sections. Specialty sections. So during the meeting, they have their own specialty section receptions where they have speakers that address that particular issue. And I put just the names of the specialty sections for you to know. So biological modeling specialty, that's probably very modern because with all this computer technology, you can really model new drugs that have the same effect, but not that damaging or not the consequences to the cells. Biotechnology, another one that is very new because it's modern. Carcinogenesis has been there for forever. I belong to that. Cardiovascular toxicology, clinical and translational toxicology, comparative toxicology, is the other different animals, and it's veterinary. There's a lot of veterinary toxicology. Computational, dermal, drug discovery, that's a very interesting one because there is a lot of drug discovery. You witness it with the COVID, right? And what is gained from that is the knowledge that could be applied to other places, right? Ethical, legal, forensics, and societal issues, because sometimes exposures, unfortunately, are so demographically unfair, and people are exposed in some regions and others are not, and exposures in the air, where are the factories located, many things like that. So there is that area also. Exposure specialty section, like on your domestic products or so. Food safety, incredible, important, food safety. See how many there are? Immunotoxicology, in vitro and alternative methods, because there is a large group of people that reject the concept of working with animals, right? Maybe, for instance, in cosmetics or things like that. So they are proponents and encourage people to use alternative methods like cellular or in silico or things like that, in which you do not need to experiment animals. Inhalation.
There for almost 20.
Mechanisms, medical device and combination products, and metals. know that one. And mixtures, we are exposed to mixtures in the air. We can study one metal, but we are exposed to a combination. Molecular and system biology, that's, well, it was founded in 1989, but I think it's also gaining weight now. Nanoscience, about nanoparticles. Nanoparticles are so toxic, but they are presenting a novel type of toxicity. So we try to understand what they do with our methods is that we have a ruler, and they do not fit in the classical traditional models of toxicology. So they present very nice challenge to learn what are they doing, and they are very, very toxic. Neurotoxicology, occupational and ocular. And more, regulatory and safety, reproductive and developmental, risk assessment, stem cell, that's 2010, right? And the last one, 2020, for millennials, sustainable chemicals through contemporary toxicology. Wow. So I think that's it for me today, and I take questions now.